All right. Okay, hey everybody, my name is Mike Lustgarten. I'm a scientist at Tufts University, and today we're going to talk about optimizing biological age. So the biological age calculator that I'm going to talk about today is Levine's phenotypic age, and what that comprises are 9 clinical variables, and that's shown here, including albumin, creatinine, serum glucose, inflammation, C-reactive protein, the percentage of lymphocytes of total white blood cells, the average red cell volume, red cell distribution width, alkaline phosphatase, white blood cell count, and age as the 10th variable. So the importance of this biological age calculator is illustrated by the finding that it's highly correlated with chronological age. So these 10 variables, its correlation with chronological age is 0.94. So a correlation of 1 or -1 is ideal, optimal, as good as it gets. So 0.94 is pretty close to 1. So it's a very strong correlation between these biomarkers with being able to predict biological age. So that data was derived from the National Health and Nutrition Study, a big data set, relatively big data set of about 10,000 people. So they replicated these findings, the phenotypic age calculator, as a measure of biological aging by looking at a separate data set, NHANES 4, and this data set included 11,000 people. And you can see visually that phenotypic age was a little bit even more strongly correlated with chronological age, with a correlation coefficient of 0.96. Now, just to put this in perspective, the epigenetic clock, Horvath's initial epigenetic clock, the correlation between that with chronological age was 0.97. So 0.94 to 0.96 is pretty close to that, if not, identical, somewhat identical. So, all right. So why is it important besides its ability to predict biological age? So the combination of those 10 variables are also associated with risk of death from all causes, also known as all-cause mortality risk. So for every 1 year increase in phenotypic age or your biological age, all-cause mortality risk increases by 9%. You can see that here by looking at all-cause mortality for the full sample. 9% increase, significant 9% increase for each 1 year increase in biological age. Now this finding was also true whether it was looking at young adults, middle-aged adults, or older adults. So there was a higher risk of all-cause mortality with a 1 year increase in phenotypic age. Now, in terms of disease-specific deaths, almost all of them on the list here: heart disease, cancer, chronic lower respiratory disease, diabetes, influenza or pneumonia, which may have some relevance to COVID-related conditions, but that's pure speculation, and then nephritis. So for every 1 year increase in phenotypic age, each of these outcomes, the risk for each of these, a death from each of these outcomes increased, with the one exception for cerebral vascular disease. Phenotypic age was not associated with that in NHANES 4. So optimizing biological age. How do you do it? So you just go to your doctor, get the standard chemistry panel, which is commonly performed at the yearly physical, plus the CBC, the standard blood chemistry count, which includes white blood cells, red blood cells, hemoglobin, and their differentials. And then you would have to ask for C-reactive protein. So the combination of those two, $75 to $80, at least in the US. It may be different in other countries. And just for comparison, so it's relatively cheap compared to other measures of biological age, including the epigenetic clock. So I have somebody crunch the numbers and so that I embedded that link on this link here. So if you want to calculate your phenotypic age, if you have all these blood test data, you can actually calculate your own biological age using the phenotypic age calculator. And knowing your biological age is interesting, but optimizing it is better. So how do you do that? So how does each analyte change during aging? What's its association with risk of death from all causes? And then what's my data? I'm going to show some of my data where applicable. Can diet, fitness, or optimization of body composition impact it? And for the last part of this, I just want to mention that I could have used large epidemiological studies to say, hey, in this study of 10,000 people, this has been shown to reduce albumin. and I actually started with that approach when I first started tracking. And I noticed that a lot of what's published in the epidemiological studies weren't affecting my biomarkers. And it wasn't until I started more rigorous blood testing and dietary tracking that I was able to find the diet pattern that best influences my biomarkers as opposed to what's been published in large cohorts. So let's see so let's start with what's my biological age. So this is my last blood test measurement 2 months ago in March. And after putting in all of my data, I come up with a phenotypic age of 31.61 years. I should mention, I think I have 8 or 9 measurements that include all of these blood test variables over the last 2 years. When I first started measuring, my phenotypic age was higher. It was actually 35 something. And I have articles on my website for anyone who wants to read about that. So in theory, you could say I've reduced my biological age over the past year or so. And that's mostly by changing my diet, improving my fitness and body composition, which wasn't bad to start with, but nonetheless, it is indeed possible to improve this in the short term. So the importance of this number, the 31 to 32 years old, it's important because my chronological age is actually 47. So I'm at least 15 years younger than my chronological age. So as I mentioned, one way that I'm reducing my biological age is through diet. So every day with a food scale, I weigh all my food and then I track that data using freely available online software. So I track everything, including macro and micronutrients. So protein, carbs, fat, and then vitamins, minerals, whatever the food tracking software can track. I log it into an Excel file. And then over the past year or so, I started tracking actual food intake. So how much of each food that I'm consuming, because to just break down food into macro and micronutrients, I may be missing a whole bunch of phytochemicals that may be optimizing certain biomarkers. So by looking at individual foods, it kind of accounts for that. So I've been blood testing about 4 to 6 times per year. And because I'm tracking my diet, I can then take the average dietary intake during that period that corresponds to the blood test, and that's one data point. So with enough data points, I can have enough statistical power to identify potentially meaningful correlations between stuff in my diet and my blood tests. Now, if you only measure once a year or less often, it's going to be harder to do that because you just won't know. It's too imprecise. So well, that's what I do. And then once I identify correlations between diet with my blood test results, I make dietary changes. I'm constantly changing stuff in my diet to tweak my diet to see how it affects and to try to push my biomarkers in the right direction. And it doesn't always do that. So that's the trial and error. That's the neural net. I use that information and then I make further changes after that. So in looking again at the phenotypic age variables, I've just posted the reference range. And the problem is most people don't know what the optimal range would be. And so what I'm going to show everybody today is that based on how these variables change during aging and their risk, how they change with risk of disease for all causes, I think we can come pretty close to knowing an optimal range. So we'll go over that. All right, so let's start with albumin. And I'm going to go through every one of the 9 variables on the list. And I want to mention that obviously for the sake of time, there's no possible way I can go through every study for each variable. So for the point of brevity, I'm going to focus on the biggest studies that have enrolled the largest amounts of subjects. These would have more statistical power to identify meaningful correlations and associations when compared with studies that may have a few hundred or a few thousand subjects. For example, here in this albumin study, this is a sample size that included more than 1 million subjects. So, okay, the reference range for albumin is 35 to 50 grams per liter. But what's optimal? So higher albumin levels are associated with biological youth, and we can see that here. So we're looking at albumin levels on the y-axis versus age. And we can see the female plot, and then we can see the male plot. So males tend to have higher albumin up until about 60 or 65 than females. So what about risk of death for all causes? Actually, before we go there. So yeah, and then going forward, you can see that albumin very clearly decreases with age for both sexes. So what about all-cause mortality? all-cause mortality risk. So this is, again, a big study, 1.7 million subjects. And we're looking at female mortality ratios on this side, and then male mortality ratios on that side. And then the x-axis goes from higher albumin levels to lower albumin levels. So how we translate this is higher albumin here, so it would be greater than 45. I have it in milligrams per deciliter. I didn't convert it back, but it's the same. So 4.5 milligrams per deciliter, or 45 grams per liter in men, and for women greater than 44 would be associated with the lowest risk of death for all causes. And then each of these different data points are for different age groups. So men less than 50, men or women less than 50, 50 to 69 years old, and greater than 70. So what we can see is that as albumin levels decrease, risk of death for all causes increases for both sexes. So can albumin levels be optimized? So I first started tracking all of my blood stuff 15 years ago, and at that time it was only about once a year. I didn't really understand the importance of testing more often back then. So based on those approximately once a year measurements for about 10 years in my 30s, my average albumin level was 4.74 milligrams per deciliter, which translates into 47. It's just a unit difference, but it's 47 grams per deciliter. So already, I don't, whatever I was doing at that time, I was already off the chart, with higher levels being associated with biological youth when I should have been somewhere around here. So whatever I was doing at that time, it was definitely on the right track. And I should say I don't have longevity DNA. The longest lived man in my family is currently my dad, who's 78. Before that, it was his dad, who was 67. So I don't have longevity DNA. I wouldn't attribute this to genetics. So then in 2015, I started tracking more often. I started dietary tracking and blood testing 4 to 6 times a year. So I have 23 data points now for albumin in the last 5 years. And what we can see is that I've been able to increase my albumin levels from the 4.74 to 4.93, and using t-test, these values are significantly different. So now I'm even higher off the chart than I was before, when I should be somewhere here, and there should have been some age-related decrease. So I've been able to resist the age-related decline in albumin. So one way I'm doing that is through diet. And when looking at correlations between my diet with albumin, the strongest correlation is for beta-carotene. And so each data point corresponds to, obviously, albumin levels. But then, as I mentioned, I track my diet, so I can look for correlations between my diet with the biomarker, in this case being albumin. So the correlation for beta-carotene with albumin is moderately strong, about 0.6. Remember, a correlation of 1 or -1, as close to those extremes as possible, is close to as linear as is a linear correlation, but it increases in strength as you get closer to 1. So granted, it's not perfect, but it's a moderately strong correlation. So I shoot for about 5, which is biological youth. And to get there, if you just go across, you can see that based on the plot, around 60,000 micrograms or 60 milligrams of beta-carotene should get me there. Now, I don't supplement with beta-carotene. For me, primary sources of beta-carotene are carrots, orange sweet potatoes, red bell peppers, spinach, and parsley. So I eat a ton of that stuff to get enough beta-carotene to potentially keep my albumin levels high. So moving forward, glucose increases during aging, and higher levels are associated with an increased mortality risk. So first, starting with how it changes during age. Large study again, 12.5 million subjects. We can see that so glucose levels on the y-axis and age on the x, and women in red, men in blue. We see that men increase, their glucose levels increase to about 60, where they somewhat plateau with a further additional increase towards later life in the 80s. Whereas for women, the increase is more linear from around 28 or so all the way up to the oldest age here. So we can see that on that lowers is better, especially when considering that it increases during aging. So somewhere around 85, 86 is found in biological youth. Now that number is important because if we look at risk of death for all causes, and that's what's shown here, the hazard ratio versus the circulating concentration of glucose levels. So the lowest point of the curve is where lowest risk for all-cause mortality would be, and for that it's for 80 to 94 milligrams per deciliter. So notice that below 80 milligrams per deciliter, all-cause mortality risk is significantly increased. And also anything higher than 94 is significantly increased. So based on this large data set, we could suggest that 80 to 94 would be optimal. So resisting the age-related increase in glucose. So again, in looking at my once-a-year measurements from 2005 to 2015, my average blood glucose level was 89. And then as I've gotten more serious and tracked more often and tracked my diet, it's 87.7. So when comparing these two groups of data, they are not different. So I basically resisted the age-related change in glucose levels, at least for now. So how's my diet correlated with blood glucose? So this is actually a strong correlation between blood glucose levels and my fat intake, total grams of fat per day on the x-axis. And this correlation is 0.75, so it's pretty strong. So what it's basically saying or suggesting is that as I eat more fat, my glucose levels go up, which is going in the wrong direction. And fat is not all just one thing. Fat's comprised of monounsaturated fats, polyunsaturated fats, and saturated fats. So they may not all equally affect or be correlated with my glucose levels. So within my fat intake, the correlation between saturated fat with glucose is moderately strong. So that may be driving this association, saturated fat. So the easy answer would be maybe to keep my blood glucose low, just eat less saturated fat. But as we'll see in a couple slides, it may not be that simple. So going forward, C-reactive protein also increases with aging, and higher levels are associated with a higher all-cause mortality risk. So the reference range is 0 to 3 milligrams per liter, but that doesn't really tell us which way, or how much CRP should be in terms of optimal for aging and risk of death for all causes. So granted, not many studies, giant studies, I mean millions of subjects have looked at CRP in terms of aging. The largest that I could find had 1,300 or so subjects, which is still pretty big compared to other studies. But nonetheless, these aren't absolute numbers too, because even though it says it's in grams per liter, the data were log transformed and back transformed. But the magnitude of the changes is what's important here. So when looking at CRP levels for men and women during aging, so looking at the various age groups all the way up to 85 or older, we can see that for men, CRP increases more than 5-fold. And for women, it increases more than 3-fold. Now, again, as I mentioned, the reference range is from 0 to 3 milligrams per liter, but that doesn't tell us what's optimal. Is a CRP of 2 just as good as a CRP of 1 or less? So let's have a look at some of that data. So less than 3, lower CRP, lower than 3 is associated with a lower all-cause mortality risk. Can we go lower? So in 3 studies, less than 1 milligram per liter was associated with reduced all-cause mortality risk, ACM risk. How about lower than that? Well, in these 2 studies, 0.86 and 0.83 milligrams per liter in these 2 studies. And we can go even further. Another 2 studies found 0.5 to 0.8 may be optimal. But it doesn't stop there. 4 studies found that CRP levels less than 0.33 milligrams per liter were associated with lowest risk of death from all causes. So from this, we can conclude, or the data suggests that lower is better for CRP, and as close to 0 as you can get may be optimal. So looking at my diet with CRP, and I have less data for CRP because I just started tracking it last year. So I think I have 8 data points, 2, 4, 6, yeah, I have 8 data points. And the strongest correlation is with my average daily fat intake. So in this case, higher fat intake, total fat intake, is correlated with a lower CRP on the y-axis. And even going forward within my fat intake, the correlation between saturated fat, actually this should be a negative correlation. So higher saturated fat intake strongly correlated with a lower CRP in my data. So that would suggest if I eat more saturated fat, and I get most of my saturated fat from coconut butter and cocoa beans, I make homemade chocolate with cocoa beans. So that may be a good way to lower CRP, at least based on this data, but not so fast. Because if you remember a couple slides ago, higher fat intake and a higher saturated fat intake were associated with higher glucose levels, which would be going in the wrong direction. So these data illustrate that, that it's a constant trial and error process. Anyone who professes to say off of one blood test, hey, this is what you should eat, I think that's spurious. It's a constant trial and error process. And as I mentioned, I have up to 25 blood tests, and I'm still constantly tweaking my diet to find out what's optimal across the board, not just for one or 2 biomarkers. So finding the sweet spot for that is key. So going forward, creatinine also increases with aging, which is associated with a higher all-cause mortality risk. So there weren't any good pictures for how creatinine changes during aging, but in this study of 9,000 people, again, Morgan Levine's group, they found increases, and other studies have found increases for creatinine during aging, even less directly, but relatedly. Creatinine is a measure of kidney function. So kidney function is estimated with the glomerular filtration rate, EGFR. So because creatinine is a component of that, EGFR as a measure of kidney function declines with age. There's plenty of data showing EGFR as estimated with creatinine levels decline with aging. So it's pretty well established that creatinine increases with aging, as a long story short. So what about risk of death for all causes? So in a study of 30,000 or around 31,000 subjects, looking at risk of death for all causes here versus a creatinine level, even though the reference range is somewhat wide, the mortality risk is actually with lower within a more narrow range. So what we can see is lowest risk is about 0.8 milligrams per deciliter, but as you go below that, risk increases, and as you go above that, risk increases. But when risk falls below 1, that's considered to be reducing risk. So within that range, it's 0.6 to 1.1 milligrams per deciliter, which is still a tighter range than, or more, it's a tighter range than the reference range. So resisting the age-related increase for creatinine. So pre-tracking values for my diet, my average value over 8 measurements in my 30s was 1.11. And I've actually reduced that over the last 5 years, whereas creatinine, as I mentioned, increases during aging. And again, these two groups of data are significantly different. So whereas there was more variability in my data, just as another illustration of why more data is better, there's more variability when I was tracking less often, but you can see the data is more tight, there's less range. And one component of that may be, the dietary tracking too. So, but I just popped the arrow up because in my last few measurements, I've been kind of playing with my diet, looking to optimize my biomarkers, and you can see that my creatinine levels increased. So in addition to tracking, the macro and micronutrients, I also track food intake, but I haven't done that for the whole 5-year period. I've just been doing that for the past 1.5 years or so, so I have less data for it. But within that data, I think this is 9 data points, 3, 6, yeah. So 9 data points for creatinine levels with my daily, average daily intake of eggs and cheese. I don't eat it every day, but it's the average amount every day. And actually there was a time when I increased it to see how it would affect my biomarkers. So what we see is as my intake of eggs and cheese increased, my creatinine levels went up, which is going in the wrong direction. With that in mind, I've cut back on my egg and cheese intake, and that actually resulted in bringing my creatinine levels back down. So for whatever reason, whether it's acid load or too much animal protein or what, I limit how much of the egg and cheese that I take in to keep my creatinine levels from going up. So just to illustrate, by tracking from all this data so far, by tracking daily diet and circulating biomarkers, you can identify the dietary pattern that leads to the best biomarker profile for you. All right, so most of the data that I'm going to show you going forward, or at least the rest of the phenotypic age biomarkers, I don't have as strong data from my own data with it, but wherever possible, I'm going to show that data. So next on the list is a lymphocyte percentage. And a higher lymphocyte percentage is associated with chronological youth. Now, this data may look a little screwy, superficial. This is lymphocyte percentage as a function of chronological age in a large study, 380 or so thousand people. So what we can see is for men in green, it pretty standardly decreases from 28% of all white blood cells to somewhere around 18%. So about a 10% decrease for the men. But for women, the data is obviously more variable. So it starts relatively high, it comes down in the 30s, it goes back up to the 50s, and then it goes down again, so that a woman at 80 or even 70 does indeed have a lower lymphocyte percentage than when she was in her 20s. So most studies don't actually look at the percentage of lymphocytes, they look at the absolute number of lymphocytes. that and how they are associated with all-cause mortality risk. So let's have a look at some of that data. So again, big study, 30,000 subjects or so, all-cause mortality risk versus lymphocyte levels, and we see that with the different colors. So lowest risk for all-cause mortality was found for lymphocytes between 2 and 2.6. So 2,000 to 2,600. This shouldn't be here, the 10 to the third. It's 2,000 to 2,600 lymphocytes per microliter. That's lowest risk of all-cause mortality. And then the highest risk for all-cause mortality is pretty obvious here. That's having lymphocytes less than 1.5 or 1,500 cells per microliter. So for the case of lymphocytes, and you could even make the argument that having too high lymphocytes, 2,600 per cells per microliter, there is a small increase in mortality risk with the yellow line, which would be indicated there. So for lymphocytes, you don't want to go too high, but you definitely don't want to go too low. And especially in the age of coronavirus, it seems that lymphocytes are dramatically depleted, so that people who have died from their studies and preprints that are out there, that people who have died from it have lymphocyte levels around 800. So that would put them obviously in the black, not good. All right, and as I mentioned, I don't have any dietary data that's strongly correlated. So all I can say for that is stay tuned. I'm going to keep blood testing. I'm going to keep dietary tracking, and hopefully with more data, I'll be able to identify stronger correlations. So mean corpuscular volume, also on the list. How does that change with age? And also, again, a lot of these biomarkers aren't well studied in terms of aging, in terms of sample sizes that have looked at that in studies. So this is a relatively small sample, but it has some weight. But before I go there, just of note, the mean corpuscular volume is basically how swollen your red blood cells. Are they filled with fluid, or are they smaller and have less fluid in them? So the reference range is somewhat large, 79 to 97%. And this data is presented in two ways. So this is data from the Baltimore longitudinal study on aging, where they track subjects longitudinally, repeated measures over time, up to 40 years or more. So they had a small group of people who had multiple measurements for up to 40 years, 25, 22, and 37 people. But we're tracking them. There is strength in the data because you're looking at the same person over time, repeatedly. And that'll I'll illustrate that, why that's important in a minute. So when looking at the data longitudinally, we see that the MCV, mean corpuscular volume, it increases from 88.8 all the way up to 92.4 in old. Now, looking at the data cross-sectionally, so just groups of young, middle-aged or old that were not tracked longitudinally, we see basically similar data. MCV increases during aging. So in looking at mortality risk, in a relatively big study, 36,000 subjects, mortality risk for men and women, lower MCVs, which are found in biological youth, are indeed associated with a lower risk of death for all causes. And we see that here for the men, lower MCV is as the reference, and then higher MCVs are associated with a higher all-cause mortality risk, and similar findings for the women. So for MCV, lower is better. So next on the list, red blood cell distribution width, which most people, at least for me, when I first got my blood test, you go to the doctor, you get your physical, you get your RDW, just gloss over it because nobody ever talks about it. But clearly based on it being identified as one of the biomarkers that's important for calculating biological age, it's important. So what is it? So it's essentially the range of the distribution of your red blood cells in terms of, are they excessively filled with fluid or not. So in this case, you'd have a low RDW because all the cells are approximately the same size. So the standard deviation of the MCV, which is how the RDW is calculated, is relatively small. But when you have red blood cell populations that are very big and very small, you have a larger range, and that results in a larger RDW percent. So what's optimal? Well, it increases during aging. We see that here with the RDW percentage versus age. Lots of data points. These are individual subject data, so it's a big sample size. And we can see that the RDW increases during age. What about mortality risk? So this is data from about 30,000 subjects, and all-cause mortality risk on the y-axis. And so we can see that the lowest risk of death for all causes is for RDWs less than 12%. And as you go higher, each of these each of these plots are higher values of MCV. So at least in terms of all-cause mortality risk in this study, lower is better. But we can go even bigger than this sample size of 30,000 subjects for MCV. In this case, they looked at more than 240,000 subjects, but the same trend was identified. Using RDWs of RDW percentages of less than 12.5 as the reference, you can see that higher RDWs is significantly higher risk of all-cause mortality. But 240,000 subjects isn't the biggest study that's looked at this. There's actually a study that looked at more than 3 million subjects. And for that, the lowest risk of death for all causes was for 8.1 to 12.5% for the RDW, whereas risks significantly increased above 13.1%. So for the RDW, it would seem that lower is better. So I have some data that is moderately correlated with RDW. Whether it's applicable to everyone, I don't know. For me, this may be a part of the story. How many calories I eat per day is moderately strongly correlated with a correlation coefficient of 0.5 with the RDW. So because higher calorie, based on this plot, it would suggest that higher calorie intakes would be associated with a higher RDW, which is going in the wrong direction. So I try to shoot for values 12.5 or less, at least based on the last slide. And to get there, if you bring the plot forward, it's somewhere in the 2,400 to 2,500 calorie range. So right now I currently average somewhere in the low 2,500s. And my goal is to get it somewhere closer to here, but one step at a time. All right, so this doesn't tell you what, calories can come from anything. So what could be driving this? What should I cut out of my calories? What food groups should I cut? And I identified a moderately strong correlation between my average daily fructose intake with the RDW. So the higher my fructose intake, the higher my RDW, at least based on this plot. So I don't drink sugary drinks or sodas. That's almost, well, I should say not never, but it's almost never. Most of my fructose is coming from fruit and vegetables, mostly from fruit though. So nonetheless, with the goal of, if this correlation is in fact driving changes in RDW, if I reduce or keep my fructose levels relatively lower, I'd expect to see lower RDW. So with that in mind, I've been shooting for the left side of this in terms of fructose intake. So somewhere around 70 or even less. So two more biomarkers on the list. Alkalinephosphatase also increases during aging, and higher values are associated with a higher all-cause mortality risk. So first, Levine, again, she found that alkalinephosphatase increases during aging. There are other studies that have also indicated this, but I don't have the time to list them all. And then what about all-cause mortality risk? So giant study, about 9 million people. This is a risk of death from all causes versus the alkaline phosphatase level. So the reference range is big, 39 to 117. You really don't know, if you're just looking at reference range, which way is optimal. So what we can see is that all-cause mortality risk starts to pass 1 once you pass around 48 units per liter. So based on that, one could say that as close to 48 is optimal, and resisting an age-related increase for alkaline phosphatase should also be suggested. So last on the list, white blood cells, which increase during aging, and higher values are associated with a shorter life expectancy. So this is data from the Baltimore longitudinal study on aging, where they measured the same subjects for up to or longer than 40 years. And what we can see is when looking at white blood cells, and this is the time prior to their death. So we can see that for those subjects, that white blood cells increase for the 20 or so year period up to death. And then how does that associate with life expectancy? So we're looking at survival on the y-axis versus time. And the reference range for white blood cells is large, and it doesn't tell you what's optimal. So in looking at survival, that may tell us more about what could be optimal. So when you look at 50% survival, that you can kind of predict life expectancy based on that. So when looking at that, the group that had the longest life expectancy had a white blood cell count between 3,500 and 6,000 cells per microliter, and we see that here. So starting from the time measurement, they lived about 38 years, that group that had white blood cells at baseline at that level. So what about higher level? Actually, what about lower levels? So lower than 35 are actually the worst on the list. They only lived 22 years. So making sure that your white blood cells are in this range. I mean, that's a 15-year difference in life expectancy. It's a big deal. So what about higher than 6,000? So 6,000 to 10,000 cells per microliter. So their life expectancy is better than the less than 3,500, but still not as good, obviously, as the 3,500 to 6,000. So they live about 5 years shorter than the best group. And then greater than 10,000, they live the second shortest. So they live about 25 years, but still 13 years less than the group that had white blood cells in the 3,500 to 6,000 range. So just as a quick, what can optimize it? This is just a study from Biosphere 2 where the subjects that were involved ended up having to calorie restrict. And there are other larger studies that illustrate this, but I think the picture here is really quick and easy. So they started off with white blood cells somewhere in the 7 range, and because they were calorie restricting or eating less calories than they normally would, their white blood cell counts came very quickly down to somewhere in the low 4s. And then once they left Biosphere 2, they returned to the normal calorie intakes, and we can see that it increased again. And then they self-adjusted and cut their calories again, and then we see it coming down again. So white blood cells may be sensitive to changes in calorie intake, but for me, I have weak correlations between my calorie intake, but for others, this may be a worthwhile try. So as I mentioned, I posted all the reference ranges in the beginning. But after going through all the data, I'd suggest that everything I've put here, and we've talked about, these numbers as potentially being an optimal range. So just as some notes, if anyone's interested in more information about optimizing these variables, I have relatively brief literature reviews, but with obviously more citations and more references for each of these biomarkers on my website. And then it's important to note that some biomarkers may be easier to modify through diet, exercise, or supplements, fitness, or other means than others. For example, for me, I'd say that, albumin, glucose, C-reactive protein, creatinine, and white blood cells may be easier to modify versus the others. And this is a continuous trial and error process. To think that you can just do it once and have it perfect, I'd argue that that's not the case. And that's all I got. Thanks for your attention, and I'll take any questions.
Yeah, and a big thanks to the Boston Police Department. Big shout out to them. Thanks for that, guys. Oh dear, it's always the way, isn't it? There's always somebody with an alarm.
Yeah. Oh, Boston is terrible for that. It's they've got to catch or do whatever. Yeah, it's loud city. Yeah,
I would have thought it had been a bit quieter with isolation and lockdown at the moment. I don't know what the situation is.
Yeah,
in Boston.
Yeah, it's just I'm on a major street, so that's probably what it is. But so, the traffic on the major streets is going to be louder, especially if the police I live across the street from the fire department, so that's always interesting. Yeah.
So yeah, I can appreciate that. Where we used to live was right by the city hospital, so yeah, it was constantly sirens going off and all sorts. And everybody used to think I lived in a high crime area. It's just that we happened to be at the back of the next street, and obviously all the ambulances are leaving.
Yeah.
So there we go.
All right, so we've got questions.
We do.
Correlation between age and the 9 biomarkers is the same in different populations. Yeah, that's a good point. For example, the glucose data was in a Korean population, but I mean, at least for glucose, the data is similar, regardless of geography, whether it's European or North American. It's most of these biomarkers are pretty standard across geography and ethnicity. Yeah. Okay, tools for data collection. So I'm just using an Excel file, and I just log everything in that. And then I'm just using simple correlations in Excel. And in other cases, multivariable regression models, but also in Excel. So instead of just looking at correlations between individual, dietary variables, sometimes I'll add a few dietary variables together to build a model to see if the model is better than the individual variable at predicting the biomarker. So simple correlations for now. I've had talks with people to use a machine learning approach, and I've talked about this before, especially at the LEAF conference last year, if anybody wanted to help me use a machine learning approach. But the people that have reached out to me and that I've reached out to have basically said that I'd need a ton more data than just, and I say just, 1,800 days of dietary data and 20 something blood tests. So, yeah.
We do know people involved in machine learning, so we may be able to plug you in, as it were. In silico medicine comes to mind. They're doing a lot of work with machine learning, deep learning.
Yeah. I reached out to Polly about crunching the numbers between my diet and biomarkers, but I don't think they were interested at that time. And I had far less data at that time too.
Oh yes, Polly Manashina.
Yes.
Manashina.
Let's see. Calling white blood cell count a biomarker is very misleading. It's not me that's calling it the biomarker. I mean, Morgan Levine, they used an unbiased approach and identified the variables that best predicted biological age. So that's one of them. So, I mean, it's included on her biological age calculator, phenotypic age, which is almost perfectly correlated with chronological age. So, calling it a biomarker is, I don't think that's a far stretch. And I wouldn't call it a crude way to measure immune health too, because, they increase during aging, they increase during infection. Now, if you said that, from Steve, if you said that not all white blood cells are the same, then we'd have, a point of agreement, because within white blood cells, the two main components in terms of how many cells are there are neutrophils and lymphocytes. Neutrophils increase during aging, and actually lymphocytes go the other way and decrease during aging. So I also track my neutrophil to lymphocyte ratio, which I would argue would be a better metric than total white blood cells, especially when considering that they go in opposite directions during aging. But Morgan Levine and others that have used these clinical variables haven't looked at the neutrophil to lymphocyte ratio as a potential variable for biological age.
I don't know. I would say that it is actually a fair biomarker, to be honest. It is indicative of something, but, arguable point, I suppose. Did anybody in here have a question? That's what I meant to ask. If anybody has a question, do feel free to raise your hand or put it in the text. If any of us here on the call would like to ask Mike a question. If not, we've got quite a lot of things coming in from Facebook as well. So you're going to be popular this afternoon, Mike. Anybody for a question in chat? No? Nothing yet? Oh, well, there's a load of questions in the text there for you.
Yeah. So Adrian, so yeah, I did plot fat intake versus creatinine. I don't remember it off the top of my head, but yeah, I'm going to go back and look at it after we're done. I don't know that it's the fat intake though, but and I also have the potential renal acid load. So the foods that you eat, the amount of acid that your kidneys would see as a potential variable for resulting in increased creatinine levels, which would suggest decreased kidney function. I also, track that too. So, it could be that eggs and cheese, because they have a lot of variables that increase the potential renal acid load. But I'm going to go back and take another look at that and see what it is. But regardless of what the reason would be, whether it's eggs and cheese or something else, for me, all I care about is getting my biomarkers to go in the right direction. So I noticed that reducing them, they go back to the right direction. And if I increase them, then they go back to, in the wrong direction. Let's see, Tony. Yeah, I haven't looked into senescent cells. I'd say the field is probably at least a few years of including that into biological age calculators, mostly because they aren't widely tested for. So, you may have small studies, relatively small studies in terms of the amount of subjects in a study that have senescent cell marker measurements, but to have large studies like, NHANES that had 9 or 10,000 or 11,000 subjects, I don't think that data exists yet. So I think it'll be at least a few years before there's large population data on that to be able to derive, biological, or to include it as a biological age marker. Other biological age clocks that I would consider as a reference. So I do use aging.ai. I did use that, but the correlation for aging.ai with its 19 or up to, I think, 31 biomarkers, its correlation at best was somewhere in the 0.8 range. which isn't as good as obviously 0.94 to 96 of Morgan Levine's phenotypic age calculator. So I've placed most of my analysis on the phenotypic age. I got a bunch of questions, huh? When is too late to change your biological age? Never. Whenever the active tracking will improve your biological age sooner, hopefully sooner than later, but eventually. And I say that not just with my diet, but I also track my fitness levels in terms of resting heart rate and heart rate variability. These are things that also change with age. And just by tracking them, so I wear a fitness tracker, just by tracking that daily for the last year and a half, I've actually made improvements and resisted age-related changes there too. So I'd say it's never too late to start. The more you track, the better you'll be able to figure out what works best for optimizing, your engine, your car. And that should reduce biological age and risk of disease. Let's see. I mean, it's tough, with your grandmother. I mean, you'd want to make very small changes, and I would only change, obvious things like diet. I wouldn't add supplements or drugs unless she has to take medications, because of the advanced age. But simple low risk strategies, whether it's changing a certain food or eating more of another food, whether it's more fiber or more nutrient density. These are low risk strategies that could potentially yield greater rewards, or even, adding a little extra walking. These are small steps can lead to great gains. So, and I'd say it's never too late. Thoughts on calicos, looking for longevity genes. Oh man, that's I don't want to.
Please, Mike. No swearing, mate. This is a family show.
Yeah. Yeah, I don't want to say anything negative. They're doing a lot of work on model organisms and looking at longevity genes, but translatability to humans is very debatable. But I'd say that the more people looking at longevity genes, the better. Keep it like that.
Yeah, I mean, the case for longevity genes isn't fantastic. I think there were a few like Clotho or Clotho,
FOXO3A.
There's a few, and genes associated with like the sirtuins, the sirtuin network as well.
Yep.
How big a variance in age, life expectancy that equates to, a lot of people seem to think it's not going to be that significant.
But also along those lines too, let's say you don't have those longevity genes or others, right? So what are we supposed to do? I don't have longevity genes, so I guess I'm just gonna, burn my candle at both ends. Who cares? Because I don't have longevity genes. I'd argue that whatever your DNA is, in terms of your maximal lifespan, whether that's 115 or it's 95, by optimizing as much as you can optimize to get the most out of your genes, that's my approach. So I don't have longevity genes. So I'm trying to squeeze, or at least not yet. It hasn't, been evident yet in my family. So I'm trying to.
I would say epigenetics probably has a greater effect on your lifespan than anything else. The environmental factors that are influencing gene expression, rather than the genes that you've got per se. I certainly don't subscribe to the idea that there are death genes that are out to get you, because I just think that idea is absurd. I mean, we're not salmon.
Yeah, and along those lines, well, I mean, there have been papers that have shown that actually longevity is a balance between how many longevity genes and lifespan shortening genes you have, right? But I agree with you about the epigenetic aging, and along those lines, in the Horvath group, there are papers that have looked at epigenetic age and relationship with diet or exercise. Actually, it's exactly as you would predict. People who had exercised and had better physical activity had a younger epigenetic age. The people who ate more basically fruits and vegetables and that higher carotenoids circulating carotenoids, one of them being beta-carotene, which is associated with albumin, had a younger epigenetic age. So there is some overlap there too.
Beta-carotene, isn't that found in carrots and similar vegetables?
Carrots, sweet potatoes. I've cut down on the carrots and replaced it with sweet potatoes to get the same amount of beta-carotene. But yeah, I could try to have more variety.
Well, now you mentioned sweet potatoes, which are really, really tasty. The only thing is, that comes to mind with those is that they're a simple carb, a bit like potatoes, and I'm sort of the thought that you need to keep your GI as sort of low as possible so that you've got a steady blood sugar.
Yeah.
And things like sweet potatoes and potatoes and similar tuber vegetables can actually cause obviously a spike in your glucose. Any thoughts on that? I mean,
Yeah. So in terms of carrots, actually, they do have a lot of sugar, whether it's fructose or sucrose, compared to sweet potatoes, which basically don't have any. So that is indeed a fair point. But carrots have a lot of soluble fiber, and which is fermented by, we're not going to get into it today, but their soluble fiber density, so the amount of soluble fiber that they have per 100 calories compared to a sweet potato, is greater. So I'd argue they may be more beneficial for the gut microbiome, but it's a 2 eggs of one or 2 eggs of the other, right? So carrots have more sugar, but also more soluble fiber, whereas the sweet potatoes have less sugar and less soluble fiber, but other things that can also affect the microbiome. So,
and it's also how they're prepared, obviously, because cooking vegetables can cause them to rise in their GI. Their GI can go up if you cook them, as opposed to them being raw. So raw carrots are okay, but if you cook, as soon as you start cooking them, whoa, hello blood sugar,
So there's another metric there, and all of us don't have the same response, blood sugar response to foods. And that study was, Aaron Siegel's group. They showed that not everybody responds to a white bread meal in terms of blood sugar rise. So whereas I may have, and I haven't done this experiment yet, but at some point I want to get a continuous blood glucose meter, stick it to my arm and see how individual foods affect my blood sugar levels. But yeah, it's variable, right? I mean, but I'd also argue that whether you're eating carrots or sweet potatoes and their potential effects on GI, these are going to be far better than eating, processed, refined foods, right? So, yeah.
So that's good news if you like your salad, folks.
All right, let's see here. Biomarkers to reduce cross-linking. That's such a big question. I mean, I'd argue that optimal body composition and a nutrient-dense diet. Just because, so when I think about collagen cross-linking or cross-linking in the ECM, I think about oxidation, I think about glycation, and these are things that would be minimized being basically lean and fit and eating a nutrient-dense diet. Now, if you want to ask me for more specifics on terms of, whether it's glucosapine or other senolytics, I'm not there yet, but that's definitely on my list, is to eventually jump into that. And, so I can only delay aging for as long as possible. And there's going to come a point where no matter what dietary change or how fit I am, we're going to need these rejuvenation, therapies. Yeah, that's on my list. Not there yet, though.
Well, spend your man for glucosapine. There you go.
Well, I wanted also pop in on the previous question that Tony had asked, and that was the biomarkers for senescent cells. And the thing is that in the human body, the only thing that, or one of the only things you have easy access to is, of course, the blood. So we are kind of limited in what we can look for. And senescent cells are not floating around in the blood, and there are not really any biomarkers in the blood that really reflect the amount of senescent cells in your body. You could say senescent cells secrete inflammatory factors, but that's mostly paracrine in the near environment of the senescent cell and don't really get much in the blood. Anyway, measuring inflammatory markers in the blood will more reflect other things that also cause inflammation in the blood, like, if you have any type of infections or, your gut microbiome that could, that through leakiness of your gut could induce increases in inflammation, or by dietary factors that induce inflammation. So there is not anything in the blood that you can measure that really reflects senescent cells easily. And kind of the same applies to glycation or cross-linking of the extracellular matrix. There, of course, your blood glucose levels will be important, your HbA1c level, which reflects like your long-term glucose level in your blood, will be important. But that all looks at glucose, and there are other factors that do cross-linking, like methyl glyoxal, for example, which is way more reactive and which you really can't measure in your blood. So we sadly are limited in what we can measure as biomarkers in an easy, accessible way.
Good point.
He's right, you know.
Yeah, great point.
Mind you, he did work on glucosapine, so he's got a home advantage. You know, you worked at David Spiegel's lab, didn't you, for a while?
Yes, for 1 year.
So he is Mr. Crosslinks. So we're fortunate to have him here with us today.
Nice. All right, let's go to more questions here. So difference in subjective well-being / performance. Yes, definitely 100%. As I mentioned, at least in terms of physical performance, my strength is pretty good. I posted a couple of videos on YouTube about me doing pull-ups and shoulder presses, so I'm still pretty strong. That hasn't changed. And I've improved my cardiovascular fitness, as evident by my lower resting heart rate and higher heart rate variability. Subjective well-being, I'm always working on improving that. I think I can always feel better, but you know, I track my sleep, so I'm always trying to improve, you know, the amount of deep sleep that I get and having consistent sleep time. So I'm obsessed with optimal physical and mental health condition. And you know, there were times in my life where I ate more processed foods and less nutrient dense diets. And you know, I'm not going to say I feel outstanding. I feel pretty good. You know, everybody would love to be chronologically 20. So, but yeah, for my chronological age, I'd say I feel pretty good, and the performance is there too, right? So, but always room for improvement. Would changes in diet over 65? Well, yeah, I'd argue that yes, and that may be the group that would need dietary changes the most. I've got a grant in submission right now that's going to look at a high fiber diet in older adults to see how it affects their muscle-related measures, muscle mass, composition, physical function. So yeah, I'd argue that, and there is plenty of data that older adults eat nutrient-poor diets, and so, but yeah, there are a few studies that have looked at you know nutrient-dense, whole food-based diets in that age group. So yeah, I'd argue that older adults would need you know, better diets across the board. So for Tomas, in terms of reading more, I'd say you should, to look up some more papers on biological age. I mean, we just went through how to optimize it, but for more background, you can go to, you know, Morgan Levine's papers or other papers, Epigenetic page, Horvath's group. You know, maybe it's not pure biological age. People have argued with me. That they think it's not really biological age, but that it correlates with chronological age. Indeed, indicates that it is a measure of a cumulative measure of biological age. Nonetheless, let's say that this panel of 9 biomarkers isn't a measure of biological aging. I mean, granted, it's associated with all-cause mortality risk, so that's important. But one of the main points of my presentation was that, you know, how do these things change with age and what's associated with biological youth? So, you know, even if someone doesn't want to identify them as a quote unquote biological marker of age, how your circulating markers of health change during aging and with disease risk, and optimizing them so we get closer to biological youth and lower disease risk. So whatever you want to call that, biological age marker or what, I'd say that's important. So, so whether that's an indicator of health span, you know, yeah, okay. So in terms of inflammation, so for Steve, I only have 8 or 9 CRP measurements right now. So, you know, even though it seems like a lot of data, it's easy to have very strong or very weak correlations. So stay tuned on how my CRP associates with other biomarkers, but I have up to 25 measurements for glucose, albumin, and the standard chemistry panel. So yeah, I do indeed look for correlations within stuff that's usually found on a blood test to potentially give me more insight at times to figure out how I can optimize them. Let's see, Steve again.
Well, it's not actually all me, to be fair. It's me typing it in the chat. I'm copying and pasting it from Facebook, and I'm putting the person's name at the front. So it's definitely not all me. I haven't asked all these questions today, Mike.
Yeah. I see it now. Right, my fault. So yeah, I'm a big fan of Greg Fahey's approach in that he wanted to optimize biological age, but not to sound negative, just to be objective, they achieved only a 2.5-year reduction in biological age using that combination HGH, metformin, et cetera. Just as, just from last year, so last year, I think I had 3 or 4 phenotypic age measurements. And as I mentioned, I started at 35 something years old, 35.4, 35.6 when I was 46. So already I started off 10 years younger than my chronological. And just looking at the 4 blood tests from last year with the 2 or 3 that I have this year, I'm 2 to 3 years younger. And purposefully, I've reduced my body weight, I've changed my diet, optimized my fitness. So you could make the argument that you don't need, for someone that was already biologically younger than their chronological age, you can make further gains by changing lifestyle-related stuff of the same magnitude that the drugs used. So, but the point about thymus involution is a good one. Steve and I talked about this before too. It's a huge, especially in the age of coronavirus, getting that thymus rejuvenated and cells that are able, like, whether also hematopoiesis, so we've got more immune cells, whether they're T cells, B cells, or other lymphocytes to fight off infectious agents. It's essential. So that's $1 billion question. If we had the answer to regenerating the thymus and boosting immune cell production that can help our antiviral defense, I mean, it would be everywhere. But it's in my mind, So the vegetarian data is tricky because I've seen data that goes both ways. I've seen data that compares vegetarians to omnivores, and they're the same in terms of outcome variables or not. So I think it goes deeper than just, what your dietary ideologies. I should say, for Mariella, that, I tried going vegan, almost pretty close to raw vegan. I presented this at other times in other presentations. So I went raw vegan, essentially pretty close to it for about a year. And my triglycerides went up and my HDL went down. So these are things that are going in the wrong direction. These are basically age-related changes that I basically used diet to cause. And then once I added fish back in, those went back to normal. So clearly being vegan isn't optimal for my biochemistry. And then even going forward, I've played around with how much beef, cheese, and eggs to add in. And then, so for me, it's minimal, once a week, once a month. But for others, maybe they would have better biomarkers on those diets. But then also, interestingly, red blood cells decrease with age, which is a problem because if you have less red blood cells to carry hemoglobin, to carry oxygen, you're going to feel exhausted all the time. You're not going to have enough oxygen to perform cellular work. And my red blood cells for a few years were trending downward, and I wasn't able to see values that I had seen 15 years ago. So I was almost, admitting to myself, well, I'm trying everything I can do, and I don't see anything improving this. What am I going to do? I guess I'm on that road for age-related decline. And for whatever reason, I added full-fat plain yogurt into my diet, and whether that caused it or not, I haven't seen red blood cell levels that are lower than 5, which is what's found in biological youth. Whereas for 15 or so blood test measurements, I was never able to get them out of the 4-7 range. So, so it's variable. It may be variable for everyone. I don't want to say go vegetarian or vegan or omnivore or carnivore. I mean, if you've got blood test results across the board, not just an isolated biomarker, not just your glucose levels are great or your TG to HDL level is great. If you can show across the board that your biomarkers are good and you have low inflammation and a low biological age, whatever that dietary ideology may be, go for it, I'd say. Let's see. The best foods to support the microbes producing short-chain fatty acids. Fiber, soluble fiber. And to most people, that conjures up an image of whole grains, and that's not true. And the reason that that's not true is because if you look at the soluble fiber content for whole grains, as they're listed by serving size, they have more than vegetables. But vegetables are more nutrient dense, but calorie poor. So for the same 100 calories, if you compare 100 grams of whole grains and pick your whole grain, oats, rice, barley, whatever it may be, 100 grams of your whole grain versus 100 grams of vegetable, the vegetable has more soluble fiber. The problem is almost no study, well, only a few studies have looked at very high vegetable diets in terms of their ability to increase short chain fatty acid levels. And I'm actually working on a grant now that I'm going to submit in June that goes after that idea in older adults. But if you're looking to support your microbes, high fiber diet. But even there's some variability. At one point I was averaging over 100 grams a day. I've been playing around with 80 grams a day to see how that affects some stuff. And I may go back to a very high fiber diet, but even that's variable. Finding the fiber amount that works best for you, is individual. Do all genes affect lifespan? That's a very simplistic. All genes are not necessarily involved in metabolism. I hate to say it. Have you seen the biochemistry chart with all the interconnected pathways? I mean, that's what I think of as an answer. It's so comprehensive. But what I just said would be an argument for not using 9 biomarkers as an index of biological age. But the argument against that is the 9 biomarkers that are on Levine's phenotypic age calculator have been studied in terms of aging and disease risk for 50 to 100 years or more. Whereas, the human genome was first sequenced in 2001. So we've got about 20 years of data on genes and even less data on epidemiological studies for genes that are associated with aging or lifespan, and even disease risk. So, I tend to favor the data. There's way more data on what's on the phenotypic age calculator in terms of how do these variables change with age and disease risk compared to the genes, which could be all over the place or not having any data yet, actionable data yet. Gut microbiota, have not checked my own in about 2 years maybe. Ubiome closed down, so I haven't done my own since then. But it's tricky with the microbiome because I think it's easy to get caught up on, is my microbiome optimal, without looking at the blood test stuff. So for now, I'm completely focused on my circulating biomarkers. But if I found something that was resistant to change in my circulating biomarkers, then I may want to look deeper. Go to the microbiome, or go to even individual nutrient levels like circulating folate or circulating something to get more insight. To do a metabolome in the blood. But so far, I don't think I need to do that. I was never really looking at the link between the gut microbiota and biological age. A big part of my hypothesis is that the microbiome does have, and there's a lot of data that supports it, that the microbiome has a big impact on aging. I'd like to say I'm on the right track because I see the chronological age being relatively low. So there are other people are looking at the gut microbiome as a predictor of biological age, but I'd like to see their diet and their physical activity levels. These are all things that can affect the gut microbiome. So I think we definitely need more studies on that. It's a dramatically understudied topic. Blue zones. So there's this the blue zones give incredible hype to the idea that centenarians in part get there by nutrient-dense diets. And I hate to say it, but that couldn't be further from the truth. There are some longevity-related societies, in Italy, for example, that do eat more fruits and vegetables and whole grains than other long-lived societies. But from my study of the literature, I'd say the older you get, the worse the diet is. And I'd argue that most people who get to 100 or 110 don't get there because of their diets at all. But because so many aren't getting there based on their diet, that would potentially leave room for improvement. But the idea of the blue zones that, let's eat these nutrient-dense diets, I'm all for it. And that doesn't mean I'm vegan or vegetarian. More nutrient density, it can't be a bad thing. Now, it could be too that the blue zones are eating less processed and refined foods, because if we're eating more nutrient dense and you've cut out the junk, that could be a contributing factor. So, how much longer would we all live if we cut down on the junk food and eat more nutrient dense foods? I mean, longevity and location. I don't think longevity. I know people can argue that longevity has an impact on it. I mean, if you're living in the North Pole and you're freezing to death every day, and maybe, but
I don't know. I mean, there's data to support heat shock proteins, Mike.
yeah.
So if you are getting frozen every day but not dying, you might even live longer.
Yeah, could, right? But that's variable, right? How about this? If your ancestors evolved for thousands, if not hundreds of thousands of years in Europe, but now you live in a temperate zone, like, so your genes are adapted to the colder climates, but now you're living in a climate that your genes are not adapted for, that may reduce your longevity. And conversely, right, if you evolved in a warm temperate climate and now you're in a cold environment that your genes are not evolutionarily adapted for, that I'd argue it may not be, but it's so much speculation, right? I have seen people, I have seen studies talking about the impact of geography on longevity, and there are indeed villages where it seems like longevity genes are concentrated, but as a broader geography, like a country, I'm not so sure. Latitude, longitude, not so sure. Now, if you showed me that SARS-CoV-2 had a greater impact in the winter in the northern latitudes and a lesser impact in the southern latitudes and in the winter months, okay, maybe, because that would directly impact your longevity. So, but I mean, it's speculation at this point.
I mean, there's certainly some data for using saunas and things. I saw Ron Patrick was covering that very recently, saunas, and I've got a feeling hot tubs as well. So
yeah,
again, you know, that's the heat shock proteins, and there's some good data for that.
Yeah.
So I think in Finland, don't they have the sauna, and then they sit and sweat for an hour, and then they go outside and roll in the snow.
Yeah.
What about that?
I mean, I've seen the sauna data, and I'm not knocking it at all. But so exercise, when you exercise, you get your core body temperature up, and if it's hot enough, you'll sweat. So you're getting your heat shock proteins activated. Now, is the sauna data in sedentary people of a certain BMI who don't exercise, who do exercise? I think there's a lot of factors there. Now, if you showed me data that the sauna reduces inflammation and improves these biomarkers, I haven't seen that data yet. But I mean, it's a low risk strategy, right? But even the opposite side, the cryotherapy for 3 minutes, standing in the minus 80 or colder for 3 minutes. I'm not sure that stuff is doing anything, regardless of, whatever few publications that are out there. I mean, these things are a cold shower. I can see that, evolutionarily, we may have, jumped in cold ponds where we've been doing that maybe for millions of years, but minus 200 degrees Fahrenheit, how is that evolutionarily accurate?
Yeah, I don't fancy that, to be honest.
Yeah,
but we are getting a hot tub, I think, soon. So I could do a one-person experiment, monitor my bios.
Sounds good. Yeah,
sounds very good, actually.
Post the pre and post blood test data, or it may be even short term, right? Maybe sauna improves glucose response, where you're better able to metabolize glucose. It could be just short term acute effects of it.
Yeah, and speaking of how to monitor that, Iris Jacobs has asked on Facebook, what are your thoughts on wearable monitors?
I have my own. I'm not going to shout out who, because they don't sponsor me, but if you see my wrist, you can probably guess. I'm a big fan of wearable metrics, wearable monitors. Mine does resting heart rate variability, and then sleep stages. I don't think there are other products that do more than that in terms of fitness variables. Maybe they'll do more than that in terms of you can listen to your music or something, but I don't need any of that. And since I started tracking in August 2018, so I have about a year and a half of tracking my cardiovascular metrics, the resting heart rate variability. And like I said, I changed my whole workout approach. I was training like once every 3 or 4 days for 2 or 3 hours at a time, lifting heavy all the time, and I was killing my my cardiovascular health metrics. My heart rate variability went down. Resting heart rate was higher than it is now. So yeah, that I'm tracking my fitness, I'm tracking my diet. I have a blood pressure cuff, I track that too. There may be stuff in my diet that affects it, so I'm constantly tweaking that to optimize the blood pressure too. So yeah, I'm trying to track as much stuff as possible.
So yeah, although Iris is apparently talking about ones that can actually assess blood composition.
Oh yeah, we're not
that's sort of, that's getting into hardcore proper medical devices, rather than, you know, like the fitness and lifestyle wearables. Although they are getting more sophisticated as well, aren't they?
Yeah, but I don't know that the wearable would ever get. I mean, when you take a blood sample and you can do a full metabolomic analysis on that, for a wearable to do that, I mean, maybe you've got a few, you know, biomarkers that the wearable could check. I mean, glucose would be relatively easy, but to have the full panel, you know, to do a full metabolome, to measure, you know, hundreds of chemicals like you could do with a blood sample with a mass spectrometer, you can't have a mass spectrometer on your wrist. It doesn't exist yet. Maybe at some point in the future, right? But no, not even close.
Yeah, I mean, how I would envisage that it would be done would be that you would ingest or implant somewhere within the bloodstream some kind of sensors, you know, for glucose and various things, and then that would wirelessly link up to a monitor either on your wrist or a nearby computer. I can honestly see that being done, and you know, achievable in the next few years. So you could then look at your metabolome in real time, which would be really interesting.
Yeah, but then you'd also have to make sure that whatever you're swallowing wasn't adversely affecting, you know, and it was benign in terms of you affecting your system, you know, your systemic physiology, right? I don't know how easy that would be. I mean, I like the idea though. Obviously, I like the idea. Yeah,
I mean, you know, it's all part of the quantifiable self. So if you've got real-time telemetry coming in, you can adjust it for maximum performance.
Yeah,
it's a bit like the Matrix, really, isn't it?
Yeah, I like the thought. Yeah, I mean, you would think the idea of quantified self, you'd see more people tracking their, you know, systemic physiology or circulating biochemistry, and I hardly see any of that. I see people tracking their mood and, you know, work productivity. I mean, anyway, I guess I want the world to track, you know, to track their metrics of health, improve the health of the world, the physical, anyway. Yeah, enzymes, huh? I mean, I don't even know how to start there. Yeah, I haven't focused on bloodletting for Dan Doherty. I guess there's some data where if you donate blood, that can reduce your circulating iron levels, which can be beneficial for health. But in terms of longevity, I haven't looked into that. And then in terms of the enzyme question for John, I mean, that could be a genetics question, right? If you have a gene for a certain protein or certain enzyme, and someone's got a, you know, one letter difference in that gene to have a different protein that has a slightly less or more functional effect. I mean, I guess that gets at that. So that's a genetics question.
Well, I mean, I'm not an expert on enzymes, but I think that the majority of the amino acids in a protein don't matter that much. It's of course the active site where it matters a lot. And if you make a mutation there, it will change the functionality of your enzyme quite a lot. But in most cases, unless it of course interferes with the normal folding of your protein, it has very little effect, most of these mutations. And of course, it is possible to make a mutation in an enzyme that will make the enzyme better, but of course that also depends on what does better mean, of course. Is it necessary for the enzyme to have a higher rate of catalytic conversion of your substrate? Is that necessary, right? I mean, is that better? Maybe in some cases, in other cases it might not be better. So there is no general answer to that question.
And then if that enzyme is at the center of a node that controls the activity of hundreds or thousands of other proteins, okay, making changes in that one protein one way or the other, right, like positive or negative, could impact hundreds or thousands of proteins, but I don't know how many proteins that would be true for, right? Like maybe mTOR, if you mess with mTOR. I mean, there are a few proteins, but there are so many animal models where you knock out a gene or overexpress it and it doesn't do anything to the systemic physiology, so, but yeah, I get your point.
And even mTOR's not completely as straightforward as some people think it is as well. So, you got TORC1 and TORC2, the two different complexes, and everyone was assuming that 2 was bad and 1 was good, but it's too reductionist, according to some new data coming in to do with rapamycin. Again, it's absolutely not clear, and this is the problem when you go so far downstream and you start tinkering with metabolism. Nothing at all works in isolation. So it's very hard when you drill downstream to actually say whether anything's definitively good or bad. And it's all about, I mean, one thing I learned from Irina Comboy was everything has the potential to be harmful or beneficial depending on the context and the levels at which it is. So like TGF-beta, for example, everyone goes, oh, TGF-beta is bad, it's bad. Yeah, but you need it, because if you didn't have any, you'd be in real trouble. So it's all about calibrating, and it's the further you drill down into the details, the more difficult it gets to control it. So
yes, true point. True.
So we got one about magnets here. Mike, any thoughts on that? Filtering blood out with magnets? I've seen that one.
I don't know about filtering it with magnets, but there are labs that are attempting to filter out pathogens, and I'm all for that. I mean, there are a few studies that have shown that microbial burden in the blood goes up with age. So whether it's magnets, I mean, if there are good studies that show that magnets can do it, or some other device, I'm all for that.
It's the idea of blood filtering is a solid idea, filtering out unwanted elements. I'm going to talk about Irina Comboy again. She's obviously her and Michael are working on calibrating various regulatory molecules in the blood, like master regulators, TGF-beta, TNF-alpha, and IL-6. Few of those. There's a handful of things at the top of the pyramid that she thinks controls a lot of stuff underneath. So her idea is to actually use, it's a little bit like dialysis, and I think it's plasmapheresis. I can't say that word, but they feed it through that's it. They feed it through a membrane, and it's able to isolate these particular molecules that they don't want. And of course, in animal studies that they've done it in, they've demonstrated rejuvenation. So there's every reason to think if you pick the right things and calibrate them correctly, you could result in the result could be rejuvenation. And same with senescent cells. If you could fish those out of the bloodstream, or the SASP factors, and tamp them down to the level that they should be at when you're younger, again, one would expect rejuvenation or resumption of tissue maintenance. We know that happens.
The only issue that I have with that approach is that that assumes that why they were produced, why those molecules, whether it's TGF-beta or other molecules, why they were produced in the first place, what's the root, right? So, I mean, I see some of that with the COVID story, people talking about the cytokine storm. Let's treat the cytokine storm. What's the root, right? The root is the cytokine storm is there to signal to the immune system to make more immune cells, but because the person can't make more immune cells, it's a positive feedback loop of a higher cytokine storm. So, I find it interesting, especially at least even in animal models, that microbial burden in the blood increases during aging, whether it's LPS or NDP. So I find it interesting that, the Comboy group hasn't decided to filter out microbes or microbial products yet. And potentially that would lead to reductions in some of the, inflammatory molecules, whether it's TGF-beta or inflammatory molecules in higher amounts that they should be there.
Mm-hmm.
And it's also just impractical to do this type of filtering devices. I mean, you know, because whatever you're trying to filter out will increase normally again if you don't take away the cause of it. So then every few weeks you will have to go to hospital and lay to a machine that filters your blood for hours on end. And there are people who do that. It's being done, plasmapheresis, for example, for people who have very high levels of lipoproteins, certain lipoproteins, when they have inherited hyperlipidemia diseases. But I mean, it's very impractical. It's not pleasant to go to hospital and lay there for so many hours, once or twice a month. And it's also very expensive, obviously.
Good point. Good point. My average blood pressure, so going back to Adrian, my average blood pressure is 120 over 70-ish, which can be better. So yeah, I'm working on that. What about ketones? I don't track ketones. So the ketone story goes back to, there's a lot of ways to fry a fish. And in terms of ketosis and longevity, so yes, a high fat, I've seen data on a ketogenic diet improving lifespan in mice, but the magnitude of the lifespan on ketogenic diet in mice isn't what was found on just a CR, just at, you know, comparing a CR group versus ad lib without being in ketosis. So, but in terms of the ketosis story, I mean, if you can show that you've, you know, got low inflammation and all the other biomarkers on a, you know, standard chem panel and CBC are good and youthful, I mean, you know, there's a lot of ways to skin a cat. So if getting into ketosis and eating a high fat diet works for you along those lines, then, you know, I'm all for it. But I haven't measured ketosis to answer Richard's question. But I bet that I have a lot of ketone bodies because eating a high fiber diet leads to a higher production of the short chain fatty acid butyrate. And that's one step away from hydroxylation to form beta hydroxybutyrate, which is a ketone. So yeah, there's a lot of ways to get to the same endpoint.
Yeah, we all like butyrate because obviously cells like colonocytes, they get about 70% of their energy from that, from those short chain fatty acids. And I know that some people try and claim that leaky gut, they suggest that leaky gut is woo and, you know, it's pseudoscience. I actually think the picture's changing on that as the data comes in. You know, the actual membrane integrity of the intestines is definitely compromised as you get older.
Yeah.
And there is considerable data now to suggest that the changes in the bacterial populations in the microbiome are very instrumental in that happening. So like the colonocytes that rely on butyrate and, you know, those sorts of things. So it's the microbiome, it's been overlooked for years, Mike, and it's now starting to get the recognition it deserves. Is it the origin point of inflammaging? Yeah, I think there's a good case for it.
Yeah, I wouldn't say it's the origin point, but it's definitely a contributing factor, right? Because I know we've talked about this before, but even in the absence of a microbiome, germ-free mice aren't immortal. So there is endogenous cell aging that's independent of the microbiome, right? So, yeah. But as Don Bodish's paper a couple of years ago showed, you know, there's a vicious cycle. So the gut microbiome changes there. So when you consider the germ-free finding, right, that there is indeed endogenous cell aging in the absence of a microbiome, there are also gut microbiome changes compositionally with aging too, and that ends up leading to a vicious cycle of increased inflammation, further worsened microbiome, further increased inflammation. So, but that on top of the cellular changes during aging, you know, for our cells. So, yeah, it's definitely a component. How much? I mean, you know, even if it's only 10%. I mean, that's 10%. You can optimize your lifespan. 10% on a 120-year lifespan is 12 years. I'll take that. So, and most people aren't even considering how to optimize their microbiome. So, yeah, it's important.
It is important. There's definitely a case for it in terms of longevity as well, where they looked at, there was a study in Japan last year, I'm sure you've seen it, where they looked at people that lived over 100, and they compared a range of microbiomes, and they found that the super ages, the people at 100, 110, their microbiomes was, you know, similar to people in their 30s and 40s, which is, you know, that's significant. You know, people that tended to age well had a much more, I'm going to say, youthful microbiome, or stable, maybe that's a better word.
So they did in some ways, but the other studies that I've seen in centenarians and their microbiomes, and actually some of the metabolic products that, besides the short chain fatty acids that are produced by the microbiome, are higher in centenarians. So for example, paracresol sulfate is one of those metabolites. Another one was phenylacetylglutamine. And the study that I have in my mind, they were calling those metabolites, which are produced only by the gut microbiome. Well, actually phenylacetylglutamine is produced in conjunction with us. So we conjugate it to glutamine. So anyway, without going into the mechanism, just long story short, they're derived from gut microbiome metabolism. But anyway, they're finding that it was higher in the centenarians. The study authors suggested that these were pro-longevity metabolites. However, these are known uremic toxins, meaning if you have kidney failure, they're elevated in the blood. So how do you reconcile those two findings? I'd argue that centenarians have gut microbes that produce these things that aren't good for health, but then they have others. Probably they have better short chain fatty acid producing bacteria and more short chain fatty acids that are similar to people who are at a younger age, right? So the good outweighing the bad. But yeah, I just think it's funny because I see a lot of the centenarian studies, and they'll say things like that, where they'll call these known metabolites that are bad for health, and they'll call them pro-longevity metabolites when it couldn't be farther from the truth.
Well, you know, everyone's got their little pet subjects, haven't they? But yeah, I mean, I think if anything, it shows that there's a strong case for eating more fiber.
Yeah, definitely. As one part of it, you know, because even the fiber supplementation studies, you know, because I get that too. Like, should I just take fiber, Metamucil, or whatever? And I, you know, because I'm working on a grant, I had to do a lit review basically on, you know, the individual fiber supplements. And most of the time they don't even raise short chain fatty acid levels, which you would predict. You know, if you're taking 16 grams or 20 grams of a fiber supplement, and that's 16 grams of just, it's going to get fermented by the gut bacteria in the short chains, because that's what the gut bacteria do. And you see no effect. I mean, I think that's important, right? So, but yet on a whole food diet, with a similar 15 or 20 gram increase, you see increases in the short chains. So there's something about the whole food and the, you know, being together in a food matrix, combined with potentially other nutrients, whether it's polyphenols or what, that, you know, further maximizes the gut microbiome independent of just taking fiber as a supplement. And actually too, in mice, there's one study where they gave soluble fiber by itself as a supplement, and the mice ended up having a greater incidence of liver cancer. So, you know, this isn't happening in people, but, you know, maybe it's an effect specific to mice, but it shows that just taking a food component outside of it existing in food may not be good for health.
Oh, there is some data for butyrate supplementation. You know, there's some reasonable
but not in old
data.
people. But not in old people.
But you know, that could be a case of that the populations of bacteria that use it are not present in high numbers as they are in youth. So I mean, you've got to have the bacteria in order to process the nutritional substances, haven't you? So you can put as much fiber in, but if you've not got the bacteria that are processing that fiber, so again, that's another argument for seeding the gut with optimal microbes again. And we look, you know, I'm surprised somebody hasn't mentioned it already today, but probiotics, you know, that always comes up. Where do you stand on that? And the answer is, well, we don't really know enough about probiotics yet to really say. Fecal transplants, yeah, maybe.
So in terms of probiotics, I mean, my stance has always been get your gut microbiome sequenced, figure out what you think is missing or what you want to be there, because it's variable, right? I mean, you know, to just say, oh, I don't have this species or I don't have this species, I want to have this species, and then I'm going to add this species, they exist as a community, right? So if you're adding one, are you changing the community structure to make it better or worse? And then you've also got the factor of, you know, is the probiotic you're taking even going to survive the acidity of the stomach? I mean, so not all probiotics are designed like that. So, And then what's the dose that you would need to seed it if indeed that is something you want to seed your gut microbiome with. So I think most people indiscriminately take probiotics without even thinking about it. And then not only that, they'll attempt to seed their microbiome, and then they won't add the prebiotics that those bugs like, right? So if you're seeding it with a bacteria that likes inulin and then you don't eat inulin, well, you've basically wasted your money because the bug is just going to get passed out of your colon. So yeah, I think there's a lot of factors that most people don't appreciate when it comes to probiotic supplementation. I'm not currently taking any probiotics. I attempted to seed my own with bifidobacteria a while ago and actually got it to go up higher than it was before, but nowhere near, other, like for example, known butyrate producer, Faecalibacterium prausnitzii. I think I had like 40% of my gut microbiome was that species, whereas bifidobacteria, I got it to less than 1%. So does that make any difference systemically? I mean, I don't know, right? So, but the butyrate argument in people, I mean, besides diet isn't the only thing that can affect the short chain fatty acid producers in your gut. Exercise too, and there's not a lot of data on this, which is another reason I'm going after it in grants. Somehow exercise, at least in young adults, led to the increase in short chain fatty acid producing bacteria and the content of short chain fatty acids in the gut. So how the hell is exercise, which you're just moving your muscles, how is that affecting the composition of the gut microbiome and what it's producing? So there's only one study, that there was a paper last year where they used lactate. Did you see that paper? They used lactate and the mice ran longer.
I think I have seen that.
Yeah, so lactate is produced by the muscles during exercise. So it increases after exercise. So they postulated that it was metabolized into propionate, which is also a short chain fatty acid. But aside from that, nobody's looked at it mechanistically. I actually think that exercise, which is known to, so you've got glycolysis, and then the end product of glycolysis is pyruvate. So there are pyruvate degrading bacteria into acetate. Acetate is then converted into butyrate. So nobody's gone after that hypothesis, but I'm kicking around that idea in my head all the time, going after, so, right, that's only one side of the story, diet, microbiome, effects on health. But then the other side is, how is muscle or movement affecting the microbiome, which in turn is further affecting muscle, right? So.
It's a good answer.
Yeah.
So the basically, folks, is we don't know. We don't know yet. More studies needed. Yeah, yes, more research needed, folks. So yeah, that's a standard answer, isn't it? In academia, is we found this exciting thing out, but more study definitely needed. So
yeah,
always more study.
Iris, diet and exercise optimize the individual. Yes, that's associated or correlated with younger epigenetic age in the Horvath group and some of Morgan Levine's papers that she's on too. So, and yes, diet and exercise is definitely a bridge. Whether that gets me 10 years of extra life or 30 years of extra life, who knows? But in terms of the importance of optimizing the fitness approach, so exercise is well known. I said this before. Exercise is well known to increase average lifespan in animal models, but it doesn't increase maximum lifespan. Basically, the gold standard of increasing lifespan in animal models is calorie restriction. So when you consider the exercise data, okay, so if I live longer than the average and the average lifespan for a man is 80 years and I get to 95 or 100, okay, but I haven't gotten to 120, right? So I would need some calorie restriction in there too. But why should exercise get you an increase in average but not an increase in maximal? Is it possible that you're doing too much? Is it chronic stress as opposed to hormetic stress that's beneficial for lifespan? I mean, so that goes back to tracking the markers, the fitness markers, whether it's resting heart rate or heart rate variability. These can tell you if you're overtrained or you've got too much sympathetic activation and not enough parasympathetic activation, which is an indicator of chronic stress. So, I'm trying to come up with the dose, whether it's diet and exercise, that can maximize everything. Now, yes, it is a bridge. As I mentioned before, we're going to need rejuvenation tech. And I hope that I can jump into that field eventually. But unfortunately in science, unless you start in rejuvenation tech, like you're doing your PhD program in rejuvenation tech, then you're a postdoc in rejuvenation tech, right? I didn't start in rejuvenation tech. So I started in oxidative stress and then serum metabolomics and then the gut microbiome. So I hope I can evolve my way into rejuvenation tech eventually. But for me, that's going to be a longer journey versus someone who's actually, been studying it their whole scientific career. Yes. Does time of the day. So I've become a big fan of time restricted feeding, and I completely changed my diet in terms of what time of the day. It may not affect my biomarkers because, my biomarkers data, the blood biomarker data is all under fasted conditions. So, I haven't done the study, after I've eaten, what do my biomarkers look like? I don't know. But I can tell you that a couple of years ago, I was eating 1 meal a day. I was on the 1 meal a day diet, OMAD, right? I would fast all day. I drink green tea in the morning, fast all day, come home around 6, and then eat 2,000 or 2,500 calories, whatever my maintenance calorie intake was at that time, from 6 to 9 o'clock. And I'd wake up every morning feeling like completely terrible. And it took me a while to even figure out why that would be. And then once the time restricted feeding studies were being, well, no, I shouldn't say once, but once I became aware of them, that, they improved measures of metabolic health by having the same amount of calories, but eating them earlier in the day. I completely flipped my eating schedule to eating the majority of my calories, almost all of them, earlier in the day. And in fact, now that we're all on quarantine, I don't have to wait to eat the small amount that I have left in the day at 6 o'clock when I get home. I can eat I can finish eating at 3 or 4 o'clock now. And, it seems to affect my sleep better. I'm not waking up as much, whereas sometimes I wake up 4 times at night because, if you eat a giant salad, a 2-pound salad, you're waking up 4 times a night.
Salad. 2 pounds of salad.
Yeah. Wow. More vegetables, more fiber. But so long story short, right? So, so now I'm eating most of my stuff during the day. I have more time to use the bathroom during the day. I'm not waking up as much during the night. I can get better quality sleep. I don't feel groggy and feel completely terrible in the morning. So I'd argue time of day definitely has a big impact on at least my psychological health. But then again, if I feel terrible because I've eaten my calories too late to bedtime, that's going to affect my workouts the next day if I do them at all on that day. So it's all connected.
Reminds me of that paper I read not long ago about they did a mouse study and then they took it to humans. Their hypothesis was because mice, which are of course related to us, are predominantly nocturnal feeders, they wondered whether time restriction for humans would work. So they actually had people eating a breakfast before dawn. I can't even imagine that time in my mind. They had them eating breakfast before sunrise, and then they were not allowed to eat. I think it was 14 hours until sundown, and then they had another meal, and it improved quite a number of health biomarkers. So there you go. It seems to suggest that we are really big mice.
So that's basically the Ramadan diet, like eat before the sun comes up and you're eating after the sun. My diet isn't too far from that, to be totally honest. Usually during the week, before probably by 8:30, I've got most of my calories in, and then I'll have a small amount, basically when the sun is getting ready to go down, where I fast completely during the day now. So, but that's a little different now. Now I'll kind of graze until, I'll eat every couple of hours until, 2 o'clock or so. But then I try to shut down my calorie intake after about 4-ish. But that, it may be different for everyone. I mean, you could make a point, evolutionarily, when the sun went down, unless you can make fire, you weren't eating. You can't forage for food when it's pitch black, and you're the prey instead of being the predator if you're actually foraging and it's pitch black. So you could make the argument that when the sun goes down, shut it down, no eating, not even to wake up in the middle of the night. I don't think evolutionarily we were waking up to pee in the middle of the night because you're potential prey. So
there was a lot. Well, I mean, our Neolithic ancestors, Mesolithic ancestors, they would have been at the whim and mercy of nature, and as you say, being outside at night, not a good idea when you've got stuff like saber-tooth tigers
Yeah.
and all sorts of other night predators that are out to get you. So
Yeah.
yeah, I don't know, but yeah, and you are right. It is the Ramadan, and they actually had it was done in the UK, this study, and they had access. I think it was in London, and they had access to quite a large amount of people who were observing Ramadan, and it was very interesting. And I think things like inflammation biomarkers were reduced, and insulin sensitivity was addressed somewhat. So yeah, people used to tell me, it's funny, my mum used to tell me off and other people when I was younger about midnight feasting, midnight snacking, going sneaking down to the fridge and helping yourself to some food, after midnight. I wish I had the data to show her then.
Yeah.
I said, well, actually, science proves you wrong, Mum. So carry on snacking.
Oh, right. Well, it depends on what time that was. If it was truly at midnight, then maybe not. But if it was, 5 in the morning, maybe, right?
Oh, I don't know.
And then depending on the age too, right? I mean, I bet when you're young, when you're, chronologically young, you could probably get away with eating anytime, whenever you want, right? But then as you age, which may probably contribute to an accelerated age, right? Probably can't get away with doing that stuff.
Yeah, true. I mean, definitely when I was younger, I can distinctly remember, you could go out, dance and mess about, go clubbing all night, dancing, and you'd eat your body weight in kebabs and drink so many. I didn't even want to think about how many pints of lager we drunk, when the amount of calories in an average night. I mean, good grief, the amount of sugar we must have consumed.
Yeah.
But it didn't really affect you when you were younger, because your body was just, whatever. It's easy, and it's able to tidy itself up and maintain itself so much better. But yeah, it's like I often say, I'm not going out to no clubs now. Drinking's a young man's game, and it really is.
Yeah.
If I have a few drinks nowadays, there's no way I'm bouncing back like I used to.
Yeah.
So yeah, there's definitely age is a factor.
Yeah.
We need rejuvenation, Mike. Otherwise, we can't go and hit the clubs.
Yeah, that's a good point. It's a good point.
That's the main. Isn't this why we're after rejuvenation technology? So
I've been thinking about it all wrong. Yeah.
Exactly. That's it. We want to tear it up like we used to,
That's the truth. Yep.
But there you go. That's enough about my my taste in music. It was good. The 80s was good, but that's another topic for another time.
Yeah, true.
And but there we go. I don't know.
Optimal range.
Oh, hang on. We got one more question.
So for Joel, there is data for lymphocyte percentage isn't generally studied as a marker of all-cause mortality. So, but there was data, I think, from 300,000 subjects that showed lymphocyte percentage and how it changes during aging. So because to get more insight for the all-cause mortality data, I used the total number of lymphocytes. So you can, I guess I should have said this. You can basically take, if around 2,000 cells per microliter is optimal, I would use that to figure out what lymphocyte percentage corresponds to that. So, but I'd argue that, somewhere in the 2,000 to 3,000 range may be optimal for neutrophils. So 3,000 plus 2,000 plus a few hundred for monocytes, eosinophils and basophils. So I'd maybe argue that probably closer to 40% lymphocyte percentage may be optimal, but there just isn't data, in terms of all-cause mortality risk. Slides available for download. That's up to Steve and Leaf.
Well, I mean, it's no problem with us, is it? They're your slides, Mike. So
yeah, put them out there.
If you want us to put them out there, we can. We can probably. We can. When we'll actually be posting the video onto the website once it's uploaded to YouTube. Probably in the next few days. So look out for that early in the week. What I'll do is I'll attach the files, and we can embed them in the article as well if people want to have a look. So yeah, I don't see any reason why not. If you're okay with that, Mike.
Yeah, sounds good. As long as I get a copy of the video, because even what we're doing now, this is like podcast style. We could chop it up into clips and, you know, release it as, you know, Joe Rogan and lots of, you know, YouTubers are, you know, posting the whole video and then chopping it up into, you know, shorter clips. I think that's a good approach. So.
Yeah, you know, it takes all sorts. So, looks like we're the torrent, and it has been a torrent of questions. Quite a lot of them actually today. So I did say you were going to be popular, Mike. So
Nice.
It does look like that the torrent is now gone to a trickle finally. But I'm glad I'm glad that people have asked so many questions, and we've had, you know, good steady figures all the way through the stream. So, and of course, many more people will watch this later on either YouTube, the website, LinkedIn, or Facebook. So, you know, it'll definitely do the rounds. I'll be interested to see how many people we reach in the end. But it's great that we've had so many questions. So thanks for everybody at home watching who's asked questions. It's fascinating. And Mike is a wealth of information on all things microbial and dietary and biomarker. Biomarker, that's not really a word, but I'm going to use it because you know what I mean. So thanks for everyone joining us, and yeah, and thanks for everybody who came in on the call as well. So unless anybody here's got any more questions, I guess we can wrap it up. And big thanks to Mike for doing the journal club with us.
Thanks,
yeah,
Steve.
he done all right, didn't he? Yeah, we'll have him back, folks. I reckon we'll have him back. He's done all right today. But yeah, so this time, I mean, you've been on the journal club before with us, haven't you?
Yeah.
But this time you were in the captain's chair. So yeah, he's done great, and I think everybody at home's enjoyed it. Seeing a lot of likes and a lot of questions, and that usually means that it's popular. So yeah, we'll do it again. And yeah, so thanks everybody who got involved today. Thanks to Mike as well for doing this. And if you want the slides and such like, they will be up soon on the website. And of course, we are here thanks to the support of the Lifespan Heroes, who are our monthly patrons. So thanks to you guys. We really do appreciate it. And it allows us to do shows like this. We're also doing a panel on May the 7th. You can find the details on the Facebook events on the page where we're streaming to. And we're going to have Felipe Sierra from the NOA, Tina Woods, we've got Daria Calterina, we've got Aubrey de Grey and David Sinclair. Good grief, what a great discussion that's going to be. So that's May 7th. So if you're about and you fancy some entertainment, they're going to be talking about COVID and what opportunities, if any, it brings for the research world in the context of aging. Could there be some good that comes of it? I'm going to be honest. I think there will possibly be an upside to this, and it's not all negative. So, but we'll find out what the experts think on May the 7th. So do tune in for that as well. And as I say, thanks to the heroes. And if you're interested in becoming a hero and supporting this and allowing us to do more, check it out at www.lifespan.io/hero. And with that said, we'll see you next time.
All right,
thanks everybody.
See you guys later.
Bye-bye. Have a nice day. Bye-bye.
Bye.
Yeah, stay healthy, guys.
Bye.